Okay, I think the slides are here, but let me try because I have a video in this slide. Let's see whether the video is also audible as well or not.
Yeah, video
Can
is
you?
going. Yeah, video is going on.
All right, and you can hear the sound, right?
Yeah. You
Oh,
now here Miss Rimshali. She will host that event. She will invite you, and then we will
okay.
start.
All right, sure.
Rimsha, just wait a minute. Let me
Should
share
I
the
stop
link.
share first?
Can I start the session or not? Or wait for 2 minutes?
Just wait uh, 2 minutes, then we will start.
So let me know when to start the session.
Uh, Hi everyone. I hope you are doing great. So let's welcome all of you in first international seminar on advancements in plant sciences. So here Miss Rimshali Zaya hosted today event. So, Miss Rimshali, please go ahead.
Assalamu alaikum and a very warm welcome to everyone. I'm Rimsha Zahir Alisha and it's my honor and privilege to moderate today's first international seminar on advancement in plant sciences. This event brings together brilliant minds, researchers, and professionals who are working tirelessly to address global challenges in agriculture, food security, and sustainability through cutting edge plant science research. To begin our seminar, I am delighted to introduce our distinguished first speaker, Dr. Nadiya Akmal. She is a renowned researcher from the University of Putra, Malaysia, with profound expertise in plant biotechnology and genetic engineering. Her research focuses on developing resistant crop varieties, a critical area in today's world where you face challenges such as climatic change, pest infestation, and increasing food demands. Through innovative applications of plant biotechnology, her work contributes to creating crops that are more resilient and sustainable and capable of feeding future generations. Dr. Nadia's dedication to advancing plant sciences has not only contributed significantly to academic research but also provided practical solutions for agriculture and food security. Ladies and gentlemen, please join me in welcoming Dr. Nadia Akmal, who will be sharing her insights on the role of plant biotechnology and genetic engineering in developing resistant crop varieties. Dr. Nadia, the floor is yours.
Okay, thank you so much, Miss Rimsha. Thank you so much, organizer, Mr. Ulam. A very warm welcome to all of you. Assalamualaikum warahmatullahi wabarakatuh, and a very good afternoon. From where I at, it's 3 p.m. In the evening. So I'm not sure where you are now. Probably a very good morning, very good afternoon. You probably a very good evening as well. So I'm Nadia from the Faculty of Biotechnology and Biomolecular Sciences. University Putra, Malaysia. Inshallah, today I'll be sharing some of the insights from what we did in our research group as a banana research group with our main focus in developing and integrated approaches for developing fusarium tolerant banana cultivars. First of all, thank you so much to the organizer. And let me share my screen in a full screen mode. Okay, can you see my screen? Is it clear?
You are
Okay.
sharing integrated approaches for development of fusarium tolerant banana.
Yeah, right.
We are seeing that
That's
slide.
right. Okay. So for my I have moved to the next slide. So in this sharing session, I would say I hope that it's going to be a very interactive session. So if you have any questions, probably Miss Rimsha, you can just ask or convey the questions from the audience. Even though I'm still presenting, I'm okay to just stop for a while and entertain some of the questions that you might have. But before I move on to the main focus of this sharing session, I will just, briefly introduce the organizer for today's session. I'm sure there are, first timers to this seminar. This is a seminar organized by Vitanova International Alliance for Science. So if this is your first time joining this kind of event from Vitanova, so I will just briefly introduce what is Vitanova International Alliance for Science before I move on to introducing the landscape of agriculture, especially banana research in Malaysia. And the main focus or highlights for this presentation would be what we did in our lab so far in our banana research group, with the main focus, as I mentioned, to develop fusarium tolerant cultivars, banana cultivars. Okay, there are four main focuses that I will touch on later. And if we have time and if the time permits, and maybe I will just briefly introduce about UPM and also my faculty. Okay, all right. So I'm not sure if you are first timer here and this is the first time you have participated in an event organized by Vitanova. So you might be wondering what is Vitanova International Alliance for Science, or the short form would be VNIAS. So it's actually an international scientific organization with a very good mission to support us, like science students, researchers, mostly from or those belong to the life sciences field from developing countries, and what kinds of supports that VNIAS can give you. They are actually doing what they are doing now, which is to provide various virtual opportunities such as conferences, training workshops, seminars. They also are very active in the social media, including LinkedIn. So later, if you have chances, you want to join some of their social medias, please feel free. You are most welcome to join. In fact, we also have a WhatsApp group as well, and they are very active. I know that they have been posting messages, announcements about opportunities for grants, for scholarships, for internships in various platforms. Okay. But the main focus of this organization is actually on the scientific research in biotechnology, bioinformatics, molecular biology, and some other related fields. But not to worry, because these organizations also engage in multidisciplinary and interdisciplinary projects. So if you are from any science background, you are welcome to join. We also have a website for Vitanova International Alliance for Science. Maybe I can show this website briefly. Okay.
Ma'am,
I have
sorry
to
for
stop sharing
inconvenience.
first. Mhm.
Yeah.
Yeah. Yeah.
Yeah. Please, can you
Okay, I'm trying
there
to
is
show the. Yeah, sorry.
sure, sure, please continue.
Okay. Right. Okay, so this is the website. So if you're interested to know more about VNIAS, so you can visit this website. I think maybe the organizer or the uh, moderator can share the link for this website. So basically, if you visit the website of VNIAS, you will get much more information here, because here there are the collaborations activities that the organization is currently doing, and also the team members. And if you have any questions, you can also go through some of the questions here. Probably they already have the answers for you. Okay, so be sure to visit this website later. Okay, all right. Now let me share my slides back. Right. So let me just briefly introduce what is the current Malaysia's agricultural landscape. You know that Malaysia used to be, or the main driving force for the economy for Malaysia used to be agriculture back in the 1970s, 1980s. But now we are moving towards more into manufacturing and services. But having said
Uh,
that,
Sorry
yeah.
for inconvenience. Ma'am, please move your slide. We are seeing Written International Alliance for Sciences slide.
Oh, really? Okay. Let me try.
Next,
Uh, is it changed now?
no,
Not yet? Not yet. Okay. It's okay. Maybe I can just reshare the screen. Wait. Let me reshare the screen first. Okay. Sorry for the interruption. Okay. I will just reshare the screen and see whether it works. Okay. How about now?
yeah, now it's understanding Malaysia agriculture landscape.
And how about now? Is it a different slide?
Yeah, it's
Oh,
fifth one slide.
all right. Okay. Now it can be changed. All right. Okay. So I did mention just now that even though the main focus for Malaysia's economy has been shifting towards manufacturing and services, but still in Malaysia we are still prioritizing the agriculture sector. In fact, even from the pre-independence period, we already have the national agro-food policies, and the latest one is actually the National Agro-Food Policy 2.0. We call it in bahasa Dasar Agromakanan Negara 2.0. So the importance of having this national agro-food policy is that for, let's say for us researchers, we know what to focus in the next 10 years, because every time the agro-food policy is published by the government, they will, forecasting what kinds of crops that we should focus in the next maybe 10 years or 15 years. For example, since I'm doing research in banana, in the previous National Agro-Food Policy, which is from 2011 to 2020, banana is actually one of the fruits listed in this National Agro-Food Policy that had already ended in 2020. So in the current National Agro-Food Policy, which is the 2.0, banana is not listed there. So it might be, we as researchers in banana might be wondering, so is research in banana not important anymore in Malaysia? Should we still produce more? And what are the reasons for this particular fruit to be opted out from the current national agro-food policy? And this could be contributed to the self-sufficiency level of banana, which is already around 100%, which means whatever we produce locally is able to meet the local demands. So this could probably one of the reasons why banana is not anymore listed in the current national agro-food policy. And as researchers, when we look further, we noticed that the producer price for banana had actually increased about 23% between 2010 to 2020. So when I look back and analyze this self-sufficiency level, what I might suggest is that this self-sufficiency level might only comprise of a few different cultivars that are popular in Malaysia. But in fact, in Malaysia, we have more than 100 cultivars. So what we think as researchers, and this is one of my personal opinions, this self-sufficiency level did not focus on many other cultivars in Malaysia that we have currently. And the other reasons that might also contribute to this higher price for banana is actually the declining in the banana supply due to the disease and also the increased production cost. I will not focus on the production cost because it is not my area of expertise, but what I will be focusing is the diseases that attack banana. Now, if you are, I'm sure you are familiar with banana, right? And you have probably eaten banana at least once in your lifetime. You know that banana used to grow happily without any concerns for diseases. However, they had been struck by a very serious fungal disease for the past, I would say, more than 100 years back. And this particular disease is called Fusarium wilt. It is a very threatening disease that affects global plantation of banana across the globe, and it is caused by a fungal disease, a soil-borne fungal actually, called Fusarium. Okay, Fusarium oxysporum forma specialis QPNC, we just call it FOC. Even though this disease was first reported in Australia back in 1874, that's why I said it had been, more than 100 years. Even close to 200 years. However, when it was first reported in Australia, it did not bring any concern to the farmers until the first epidemic occurred in Panama in 1890. So that's when people started to concern about this Fusarium wilt. And it is also one of the reasons why Fusarium wilt is also dubbed as Panama disease. Now, if you happen to pass by banana plantations or banana farmers, you might be wondering what are the symptoms of Fusarium wilt. The symptoms of this disease can be categorized externally and also internally. So if you were to observe the external symptoms of Fusarium, you will be seeing some of these yellowing and wilting symptoms on the leaves of these banana plants. However, this is not a clear sign because these particular symptoms can also be caused by other factors. So you have to also check the pseudostem of the banana, and you will see the discoloration in the pseudostem of the banana. And of course, you have to, perform some other molecular analysis to confirm the presence of this Fusarium pathogen. And what is so bad about fusarium is that once it's in the soil, it is almost impossible to be eradicated. There are some reports that mention if the fusarium is there in the soil, it can live and detain or remain in the soil up to 30 years. So you might be seeing plantations once it's struck by fusarium, they choose not to plant any banana anymore in the next plantation season. So they might other, choose to switch to other plants, or they just discard the whole plantation. Now, obviously, the impact of fusarium is very huge, not only in Malaysia but globally. I will just touch a bit on the impact of fusarium in Malaysia. It has caused around 253 million dollars loss in terms of the monetary loss in Malaysia, and there was a survey conducted by a group of researchers in University Malaya in 2022 that reported almost 60% of the smallholder Malaysian banana farmers affirmed or confirmed that their plantations had been attacked by either fusarium, which is the focus of the study, moko or sigatoka, at least once in the past 5 years, showcasing the seriousness of this disease in Malaysia. And globally, um, I'm not sure. Maybe you are familiar with a Cavendish cultivar. So this is the main banana cultivar that is traded or being traded globally. But before we had Cavendish, actually the main cultivar that was traded in the 1940s, 1950s is actually Gros Michel. Okay, it's called Gros Michel. However, due to the attack of fusarium, Gros Michel had to be replaced by a new variety called Cavendish. Unfortunately, Cavendish is also susceptible to a new variant of Fusarium. It's called Fusarium TR4. So what we are seeing now is, even though we can have certain cultivars that are resistant to Fusarium, but if the Fusariums evolve, then we might risk losing that cultivars as well. Hmm? Okay, wait. Okay, sorry. My kid is asking for the chair. All right. Okay. Now coming back to what I did in our lab. So since I mentioned that we have more than 100 cultivars and banana varieties in Malaysia, we thought that why don't we shift the focus to explore the genetic potential of these banana cultivars and also the economical potential of
Uh,
our
Hi, ma'am.
local banana.
Sorry for
Yeah,
inconvenience.
yeah.
Uh, is you explaining fifth number slide?
I think I have changed the slide. Is it not working?
Uh, we are seeing fifth number slide,
Fifth
five number.
number. Five number. The five number. I'm not sure why my slides are not working. Mm,
Which
so I
slide
have
you are?
changed a few slides. Uh, are you still seeing the same slide? I have changed my slides a few times.
Right
Do you
now
see?
here, fifth number slide.
What is the heading for this slide?
Yeah. The national agro food. Yes. Now it's new.
Really? I have changed a few slides. I'm not sure why my slides does not change. Okay, I have a PDF version. I think I can just share the PDF version. Okay, let me stop sharing first. I will just share a PDF version. Okay, sorry for the technical issue. Okay, let me share a PDF version of my slide. Okay, I will try to reshare my screen. Okay, let's see whether or not this works. Okay, this is a PDF slide, not a PowerPoint slide. Okay, I have changed my slide. Can you see it?
Yeah. Now, please, can we start from here? Just overview. Yeah.
Okay, all right. So I stopped. My last slide was here. Is it?
Yeah. Yeah.
Sorry for the technical issue. I have actually changed my slides a few times, so I will just briefly go through some of the slides. So previously, when I talk about the banana production in Malaysia, so this is my slide, and this is the slide when I introduced the fusarium wilt and mentioned that it was first reported in Australia back in 1874. However, the first epidemic when the people get really concerned about fusarium is actually when they had the first epidemic in Panama in 1890. So we had a very long history with fusarium wilt about more than 100 years back. Okay, and this is where I show the symptoms of fusarium. So you did not get to see these slides just now, right? The banana symptoms on the leaves and also the pseudostem. This was not
Yeah.
also shown just now. Okay. All right, I'll just go briefly go through this. So I mentioned just now, if you were to observe the symptoms of fusarium wilt, you can focus on the external symptoms and also the internal symptoms. So for the external symptoms, you will be seeing the yellowing and wilting symptoms on the leaves, but since these symptoms can also be mistaken by other symptoms caused by other diseases, so you have to also look at the symptoms in the pseudostem. So you will be seeing the discoloration of the pseudostem, and also of course you have to mention or conduct the molecular analysis to confirm the presence of the pathogen. Okay, all right. Okay, let me get this blanket for my kid first. Okay, one more. All right. Okay. Okay. So I had mentioned just now about the seriousness of this disease. Once it's in the soil, the fusarium pathogen, it will be almost impossible to be eradicated because it can retain in the soil up to 30 years. And just now I did also mention about the impact of fusarium wilt. So this is the impact of fusarium wilt when I mentioned about the survey that we conduct among the Malaysian banana farmers. These are the slide for the survey that I mentioned just now. And what is more concerning about fusarium is that if there are some cultivars that used to be resistant towards fusarium, once we have the fusarium pathogen evolve and become a new variant, it might attack a new cultivar or the current cultivar that used to be resistant towards it. Because in fusarium, we have a few different variants for fusarium pathogen. So that's why it's very important to have a very integrated disease management strategy for fusarium, because, for example, Gros Michel used to be the main cultivar that we trade in the 1940s, 1950s, and it was susceptible to fusarium attack. And we had to replace Gros Michel with Cavendish, the one that is the main traded banana now. But currently, with the emergence of fusarium wilt TR4, TR stands for tropical raceform, Cavendish is actually susceptible to fusarium TR4 as well. So what if this fusarium TR4 attack, the countries that trade Cavendish globally, such as the Latin America, the Caribbean? In fact, they had been attacked by the fusarium. It's just that they managed to, control the disease. But if Cavendish is severely attacked by TR4, then we might have to find some other cultivars to replace Cavendish, just like what we did in the 1950s. So this is to showcase the seriousness of this disease. Having said that, in my research group, in our research group, we are trying to leverage the richness of cultivars that we have in Malaysia. Because in Malaysia, we have more than 100 local banana cultivars and varieties. So we think that they are still underexplored, and we only focus on a few commercially popular cultivars. So we are trying to exploit this genetic potential of many other local cultivars and see whether or not we can highlight this as a new cultivar and shifted our focus from the commercially cultivars that we have nowadays. And we also focus on the tissue culture landscape in Malaysia. They Currently in Malaysia, the tissue culture production for banana only focus on these few commercial cultivars. So we have to also strengthen the tissue culture landscape to address the farmers' concern. And we think the way forward is to strategize our disease mitigation strategy and diversify the production through research. Now, in our lab, we had four main focus, okay, for developing fusarium tolerant banana cultivars, and I will touch one by one using some of the findings that we had from our research conducted in the lab. The first focus is the identification of fusarium tolerant varieties or cultivar. The second one is identification of antifungal proteins in banana. We believe that this is very important because if we want to develop the third focus, fusarium tolerant banana through transgenic approaches and gene editing, we first had to identify what is the potential defense related genes in banana that we can, overexpress or knock out in developing the tolerant banana varieties. And lastly, we also had a few research in our research group to identify the potential antifungal compounds and beneficial microbe. So this is basically the proteins in banana, and these are basically the other antifungal compounds that could be coming from other resources as well as other bacteria. Okay. Now, the first focus, identification of fusarium tolerant varieties or cultivar. Um, I had actually mentioned this before. The cultivated bananas that we have currently are asexual. You know that most cultivars, if they are triploid genome, they are propagated asexually. So you have to propagate the banana through suckers. But for most wild bananas, they are sexually propagated. So the difference between the cultivated bananas and wild bananas are, of course, the genetic diversity of these bananas, because for the cultivated bananas they have low genetic diversity. They cannot do much to mix their genes, right? But for the wild bananas they have a higher diversity of genetic mixture. Unfortunately, they have poor agricultural traits. So normally the wild bananas, they will not really, eaten raw or used for cooking. So to address this, we know that the challenge lies in the cultivated varieties that had low genetic diversity. So because of that, we have a problem that these cultivated bananas, they are more susceptible to diseases. And the few resistant types that were identified, mostly they are coming from the wild bananas or bananas that had low commercial potential. So for this project, this was actually conducted by one of my PhD students. This project focus on, there are three main focus for this project. First, this project are really interested in screening the local cultivars in Malaysia using ISSR markers, aiming to find a resistant cultivar. Okay, resistant cultivar. I will show you some of the results later. And of course, we have to test whether or not the cultivars or varieties are indeed tolerant or susceptible to fusarium. So we use a bioassay platform to test the susceptibility and also tolerance level of this cultivar against fusarium. And we also conducted a biochemical validation using PAL gene. So PAL, or phenylalanine ammonia lyase, is actually an enzyme that is involved in the plant defense. And why PAL? You might ask, why do we choose PAL for the biochemical validation? Because our previous research had shown that resistant banana cultivars had higher PAL activity. And not only from our research, we also find some other researchers that reported higher PAL activity or stronger PAL expression in the resistant cultivars. So that's why we choose PAL gene for the biochemical validation. So these are the objectives that I mentioned for this project. So what we did in this project, our team went around a few different places of plantations in Malaysia, and we collected up to 21, actually 21 local cultivars. So if you look down here, these are the 21 cultivars that we collected from all over Peninsular Malaysia with different genome groups. Okay, and the students conducted ISSR marker analysis using 21 different markers. And interestingly, when she conducted or constructed this UPGMA dendrogram based on genetic similarity of these 21 local bananas, we found that there is actually a cultivar called rotan, so this part here, that evolved together or are very closely related to another cultivar called Jari Buaya. What is interesting about Jari Buaya had been reported as a Fusarium TR4 resistant cultivar. So we think that if rotan and Jari Buaya are closely related and they evolve together, rotan might also be resistant or tolerant to Fusarium. So what we did next from this list of cultivars, we choose 7 cultivars. The one in the red box are the one that we choose for the tissue culture propagation. Okay, tissue culture propagation. The reason why we conducted the tissue culture propagation is to prepare the plants for the USA Fusarium infection assay later. Okay, do you still see my slide? I'm showing the dendrogram or the phylogenetic tree. Can you see the slide? I'm afraid that my slide is not moving.
Yeah, it's phylogenetic tree slide. So
Yeah, all right. Okay, great. Okay, great. Okay, so we did tissue culture for these 7 varieties, but I will just show you the result of Fusarium bioassay from 3 of the cultivars. So I did mention a few cultivars just now. The most popular cultivar in Malaysia, the local cultivar I mean, the local cultivar that is the most popular in Malaysia is called Berangan. And we know that Berangan is highly susceptible to Fusarium. And sure enough, when we develop the tissue culture of Berangan and we infect this with Fusarium, do you still remember the symptoms that I showed you just now? So this is 62 days after infection with Fusarium. Compared to the control, so the top panel is the control and the lower panel is the Fusarium infected Berangan cultivar. Sure enough, we saw the discoloration in the pseudostems and also the wilting and yellowing in the leaves of Berangan cultivar. And this is what we expect because Berangan is highly susceptible to Fusarium. We also tested another cultivar. This is called Awak. So Awak, if you compare to Berangan, the discoloration in the pseudostem is much actually better. Okay, better than Berangan just now. The wilting and yellowing symptoms on the leaves are actually better compared to Berangan. However, it's not totally immune to Fusarium because you can still see some of this discoloration in the rhizome of this Awak. So this is Awak. Awak is another local variety in Malaysia. And do you still remember the dendrogram just now? I highlight the rotan cultivar because it is closely related to Jari Buaya, the resistant cultivar. So we are very excited to test whether or not rotan is also resistant to Fusarium. And when we conducted the Fusarium bioassay, this is the result for the rotan cultivar. And we can see that... Adik, no, Mami tengah work. Okay. So we can see that the rotan cultivar did not have any discoloration in the rhizome after they had been conducted or infected with Fusarium. Okay, and this is the control panel. Of course, they are free of infection as well because the control plants are only soaked in sterile distilled water. But for the Fusarium infected rotan, they also did not have any discoloration. So this reflect the dendrogram just now because we also assume or predict that rotan would be tolerant to Fusarium. And of course, when we scored all the symptoms from the rhizome and also the leaf, we will see different scoring because we tested 7 different cultivars. But sure enough, Rotan had the lowest disease score, okay, for both the rhizome and also the leaf. And these are actually the translation for the cultivars that we tested, because from the disease score we have to, translate the scores to interpret whether or not the cultivars are susceptible, highly susceptible, or tolerant against fusarium. So berangan is actually highly susceptible and rotan is considered tolerant. Okay. Now, next, what we did was we want to focus on the poultry gene just now. So we form a construct of poultry and transform into tobacco. We did not actually transform in the banana yet because we did not have the explant for banana at this point of research. So we just use the model system, the model plant, which is tobacco. And this is a transgenic tobacco overexpressing the poultry gene. And we tested a few things in this transgenic tobacco, such as the pulp enzyme activity, the lignin content, the phenolic content, the phenolic content, and from a few different lines that we regenerated from these transformation events, we only focus on two lines which had generally among the highest pulp enzyme activity and lignin content, which are L10 or line 10 and also line 134. So for these two lines, we proceed to the histological analysis. And you know what's interesting about PAL is that it's a defense-related gene and it is involved in lignification. Okay, so when we perform histological analysis to analyze the lignin deposition in these two lines, line 10 and also line 134, compared to the wild tobacco that had no overexpression of PAL, we can see, a higher deposition of lignin in the transgenic tobacco compared to the wild tobacco. So we assume that this deposition of lignin might also contribute to why the plants would be more resistant towards pathogen attack, because lignin is considered one of the first line of defense when the pathogen attacks the plants. So for this transgenic tobacco, since it's not a host to fusarium, we only tested against another pathogen that is called Rastonia that can attack tobacco. And we can see that from one of the lines of tobacco here, the I think it's L134. Yeah, what L134? It has a lower disease score, which means they are more tolerant towards the Rastonia infection. So this is what we conclude from the first focus, which is to find the cultivars that could be tolerant or resistant towards fusarium. So we managed to identify the cultivar rotan that we think could be tolerant towards fusarium. And the PAL analysis also showed that overexpressing PAL might improve the defense of the plants against pathogen attack. So this is what we conclude from the first focus in our lab. Now moving on to the second focus, which is identification of antifungal proteins in banana. I have a few students working in this part of the project in which we are interested in finding out which pathogenesis related proteins, or we call it PR proteins, that had antifungal property, because fusarium is a fungal. So we think that if we can overexpress antifungal protein more abundantly in the banana, then we can improve the overall plant defense of this banana against fusarium. So I had a few students working on this, and generally when we work on this kind of project, what we do first is actually some bioinformatics analysis, where we have to retrieve all the candidates from the database. We have a banana database for this work, not our own database, of course, but this is a globally used banana database. It's called Banana Genome Hub. So from here, from the retrieval, we actually analyze the expression for this particular gene before we proceed on to the functional characterization of that particular protein, and tested the antifungal assay for that particular gene or protein that we had expressed in a heterologous system. We express it in E. coli. Now, what we did so far in our lab, we had focused or worked on few different PR10, PR5, defense scene, and also PR14. And from each of these PR, there are actually more than one copy. Of course, if you are familiar with plant genomes, most plant genes I think will have more than one copies. So in banana, for PR10 we have 10 copies. For PR5 we have 46 copies. For defensin we found 6 copies, and for PR14 we have 55 copies. And these are all being done or had been conducted by few students in my lab. We have master students and also final year students working on this project. Now the next challenge is, from these multiple copies of genes, we have to determine which of these copy actually had the antifungal potential. So again, for this method or for this approach, we will use bioinformatics. So we conducted a few bioinformatics analysis, and I'm using PR10 here as an example. So from this bioinformatics analysis, such as the gene ontology analysis, the phylogenetic analysis, we will be able to um, find, or at least identify just a few candidates out of those sorry, of those many copies of the genes that we identify. Okay, let me just share one example here on PR10. How did we come to conclude out of 10 PR10, there might be only 4 PR10s that potentially have antifungal property. So this is what we did in our lab. We construct a phylogenetic tree where we put all the 10 PR10 inside this phylogenetic tree. Sorry. At the same time, we also went into literature review and find what are other PR10 from other plant species that had been proven to have antifungal property. So we also took those PR10s from other plant species and include them together in this phylogenetic tree. And from here, we will find the closely related PR10 copies that are or that evolve together or might be related to our banana PR10. So this is how we identify or shortlisted our antifungal candidates using bioinformatics. Of course, we proceed to gene expression analysis. You can use qPCR, or in my case, we use nanostring. So any methods you can use as long as the aim is to identify or analyze the gene expression. Now, moving on next, if we had identified one candidate or at least one or two candidates from the PR10 groups, then we move on to, from the construct, transform into a heterologous system. In this case, we use E. coli, express the protein, purify the protein. So these are some of the results from the expression of the PR10 in E. coli. And also this is the purification of the PR10. And once we get the purified protein, we can perform the antifungal assay. And of course, we will be testing against Fusarium, right? But if you have any other pathogens, you can test as well, because they might have a similar mode of action. So you can test against multiple pathogens. But in our case, we tested first against Fusarium. However, for this PR10, we only managed to get a very low amount of purified protein. It's around 0.2 microgram. So when we tested against Fusarium, we did not see any significant inhibition, okay, when we compared to control. However, in the future, if we had time, we might try to express more of this PR10 and test it against using higher amount of purified protein. Now, that is the second focus of my project or our lab. Up to this point, do you have any questions? Maybe we can answer one or two questions before I move on to the third focus.
Yeah, thank you so much.
No
If
questions so far?
anyone have any questions, you can write in
Yeah.
chat box. We will read it
Oh,
and we will answer your question.
yeah. If not, then maybe I will proceed first. How much time I left, Gulam? How much time do I
Uh,
have left? 10 minutes?
time, if we talk about time, 15 minutes
50 minutes. Okay, okay. All right. Okay. I'll be able to finish in 50 minutes. All right. I'm afraid that I will go over time. All right. So just a quick glance on the third focus in our lab, in which we developed the fusarium tolerant banana through transgenic approaches or gene editing. If you still remember, my second focus in the lab, which is to identify the antifungal candidates from the banana genome. If those are successful and if we have potential genes, then we will move on to the third focus. But currently, for the third focus, we only had a few, I think, two projects. Yeah, two projects. Previously, I did actually develop a transgenic banana using PR10. So you keep on hearing PR10. So PR10 is actually one of the main genes that we work on in our lab. So in my previous project that I did for my PhD, I actually overexpress PR10 in a transgenic banana. So these are the transgenic banana overexpressing PR10. And from tissue culture, I move on to the polybag or pot, where I grew the transgenic plants up until maybe 20 centimeter, 30 centimeter in height. And I infect with fusarium just to see the symptoms, okay, the development of the symptoms in the leaf as well as in the pseudostem. So this is the non-transgenic, okay, non-transgenic banana, and these are the treated transgenic banana. So this is the non-transgenic banana, but also treated with fusarium. So I can see that the development symptoms for the transgenic banana is actually slower than the non-transgenic. However, at the end, the transgenic banana are still infected by fusarium. It's just that the development symptoms are a bit slower compared to the non-transgenic. So this is the first project on the transgenic. I also have a current project on gene editing. So this is another gene. It's called CIPK, but still it's at the very early stage of the research, where we just managed to amplify the gene, and later we will move on to the construct, okay, the CRISPR construct, before we can proceed to transformation and regeneration. Okay, so these are what we did for the third focus in the disease management of fusarium. Now, the last part was actually a work done mainly by my co-researcher, Dr. Norbaiti. I am one of the co-researcher in this project as well, in which she is interested in finding the potential antifungal compounds that can, inhibit the fusarium growth, and also some of the beneficial microbes that can also be or perform antagonistic effect against fusarium. So one of the master students in this project, Priya, okay, she tested the plant extract, essential oil, and hydrosol from few different plants. Um, I think they have like tea tree oil, lime, and from here, what she found is actually the potential of tea tree. Tea tree oil is found to have some inhibitory effect against fusarium, both in vitro, because I'm actually showing the PDF slide. At the back of this particular image, we had actually an image of this in vitro assay of tea tree oil. Unfortunately, it's not visible here because it has been blocked by this particular image. However, what I can tell you is that tea tree oil is also one of the potential antifungal compound that has shown inhibitory effect not only in vitro but also in vivo. Okay, so this is the in vivo experiment for tea tree oil. So this is the non-treated plant that is only treated with fusarium but not treated with tea tree oil. And we also have a few different plants that have been treated with different concentration of tea tree oil, and we can see that the symptoms actually improve. The symptoms of fusarium in the plants actually improved if higher concentration of tea tree oil were used in the treatment compared to the non-treated plants, which showed very severe discoloration, and also this is already considered dead plant. Okay, so this is to highlight the potential of tea tree oil as an antifungal compound. And this particular project also tested a few different microbes. These are a few different microbes tested here, and some of the microbes here actually showed some antagonistic effect against Fusarium as well. If you were to compare, so this is the plant that is not treated by any microbes, but these are a few different plants that had been treated with different microbes, and the symptoms actually improved as well compared to the control that had not been treated with any microbes. Okay. Now, having said that, even though we had, routinely identified antifungal compounds, antifungal genes in our lab, there are still a few challenges remain in the research of banana and also fusarium in our lab. For example, we had a routine of, analyzing the symptoms of fusarium based on the rhizome, based on the leaf, but the challenge is different students might have different interpretation of the symptoms. Even though we have a rubric, but sometimes the interpretation can be quite vague. So we think that if we can empower the interpretation using AI, then it will definitely help us to have a faster and more accurate assessment on the disease severity. And we also had challenges in establishing and maintaining some of the tissue culture materials because students come and go. So once the students finish their project, so we might have problems to find other students to maintain and continue subculturing the tissue culture materials. So this is also one of the challenges that we face in the project, as well as of course the grants lah. Okay, the grants that we have to compete with other researchers working on other plants or even working on bananas as well. Okay. Now, I think that is all for what we did in our lab. If you have any question, you can share or type in the chat box. But since we still have time, let I briefly introduce our university, UPM. So a warm welcome to all of you. If you are not familiar with University Putra Malaysia or UPM, UPM is actually the largest university in Malaysia. Okay, we had two main campus, one in the Peninsular Malaysia, very close to the capital city, which is Kuala Lumpur, and also very close to the Malaysia's administrative capital city, which is Putrajaya. So it's like 20-minute drive to Putrajaya and maybe 30 to 40 minutes drive to Kuala Lumpur if there is no heavy traffic. So the location is very strategic. And currently we have more than 27 students, close to 28,000 students, comprising both the local and international students. And at the global standing, we are currently ranked at the 134th in the QS World University Ranking and 20th in the Asia Ranking. And we are very proud of our Green Campus as well because we are at the 24th rank in the World Green Metric University Ranking. Um, I am from the Faculty of Biotechnology and Biomolecular Sciences, uh, and my faculty is actually very close to the UPM MRT station, the train station that can connect many different stations to Putrajaya and also to Kuala Lumpur. So this is the UPM MRT station, and this is actually my office and my lab. So it's like maybe 5 minutes walk from the MRT station, from the train station. So it's very close by. And this is actually the closest faculty to the UPM MRT station. Okay. Gulam, I have a video that I would like to show you about UPM. Do we still have time? Do we still have time? Can I show a short video?
Yeah, yeah, sure.
All right. Okay. Let's try. I'm not sure whether this this can be shown or not because my slides are quite problematic just now. Okay, let me try. Let me try. Mm, okay, so is the video moving and is there any sound?
Yes.
All right, okay. So just enjoy the video. It's about UPM, and if you have any questions later, you can just let me know. Okay.
There is not any video. It's a slide.
Sorry.
We are seeing slide,
Oh,
not
not
any.
video. Not any video. All right. All right. Okay. Let me try to find the original video. Okay.
You
Let me.
can show it by sharing your screen. It will not be open in slide. It's privacy
Ah.
of
Okay.
YouTube.
All right. Okay. Let me try. Mm, okay. I will try to reshare again. Let me find the video first. Okay. Let me try one more time. I will stop share first.
Till the open video. If anyone have any question related to previous session, you can write your question in chat box, and we will provide you the answer. If
Okay.
anyone have any question, you can write in chat box. Thank you.
Okay. I'll try to play the video one more time, see whether it works or not. Okay, how about now? Is it working? I've stopped the video.
No, we are not seeing any video. No.
No, okay. Okay. Maybe one last time. Okay, let me try my entire screen. Yes. Yeah, okay, okay. Later, later. Okay, let's see one more time whether or not it works. Oh, Adik, wait. Yeah. Is it working now?
Yes, okay.
All right. Okay. Okay, bye. So I think that's all from me. So I'd like to finally thank the organizer, Vitanova International Alliance for Sciences, and of course, the researchers in my banana research group, Dr. Puat and Dr. Baiti, and all my students and my collaborators for their inputs and for their knowledge and contribution in the projects that I presented today. Thank you so much for your attention as well.
Thank you so much, ma'am. Miss Rimcha, kindly conclude that presentation.
Thank you
And
very much, Dr. Nadia, for your valuable insights. And now it is my pleasure to introduce our next distinguished speaker, Dr. Wazir Ali. Dr. Wazir Ali is currently serving as an
please,
associate.
Miss Rimcha, let me add him.
Sure, sir.
Uh,
Hello.
hello, hello, doctor. Am I audible?
Yes, yes, fine. Please share, sir, your presentation.
Yeah, I'm sharing. In while, please wait.
Hello.
Yes, sharing, please. 1 minute.
Yes, you please. Yes, yes, sir. Um, okay. Uh, Hello. Yes, this is Dr. Wazir Ali, an associate professor, Department of Molecular Biology and Genetics. Objective: the Advance in Plant Biotech, first international seminar on advance in plant sciences, first international uh, uh, advance in plant sciences, organized by NOAA International Alliance for Sciences. So I am the highly thankful. So I share my key points in the advance in plant biotech, molecular biology, climate change, insight and sustainability practices, research and academic guidance, and future collaboration. Next, please. So, first of all, I defined the objective of the seminar. The main objective of the seminar to create awareness among the students about the plant science, because the international seminars cover comprehensive and the plant science and as well as the advanced plant sciences. So first of all, to enable second object to enable the students enhance the biotechnology and molecular tools. What is the biotechnology and how the molecular tools used in the field of the plant science through the genetic engineering. Where the next key point, the impact of the climate change and their effects on agriculture productions. Agriculture production. Hello, hello, sir. So plant science.
Yes, yes, your voice is
Okay, sir. Okay. Next, please. Slide next, please. So plant science, if we look into the plant science are alive like and just like a people and animals. So if we look into the plants, the plants are just like animal and people and animals because they grow and die. They need energy, nutrients, air, and water, and they react to their environment and react to their environment, how they grows and how they uptake the nutrients from the soils, what kinds of the irrigations is required and what kinds of the various kinds of the inputs is required for the growth of the plants, and which type of the soil is required for the plant growth, for the plant growth. So plants, if we look into the plants, the plant provide us with oxygen, food, fuel, and fiber. Among other reasons, scientists have developed plans to secure, improve the secure food supply for an increasing world population. If we look into the uh, world population, the today's world population is 7.5 billion, and 2005 the data reported, and where the populations will be doubled and 2050 the 90 billion people say they need for the uh, they need for food, shelter, cloth, fuel, and others. So they need for the food, cloth, and shelters. So scientists have developed improved the secure and supply for increasing the population to develop the new source of genetic resources, new source of the genetic resource, and how it becomes a new genetic resource, because the genetic resource means the any type of the biological materials, any type of the seeds and piece of the DNA and function units of the heredity and how the conservation, because scientists have developed the different types of the methods for conservation of the genetic resources, such as 5 to 7 years and 10 to 20 years and 20 to 100 years for the future generation. Where is the scientists have a new source of genetic resource and their conservation like
sorry
agriculture.
for inconvenience, dear doctor.
Uh,
Uh, we are on
slide number plant science, sir.
slide
Pl-
number 4, right?
Plant science slide number two. Slide number two. Sorry, slide number three. Plant science. Plant science. Next. Okay, sir. Okay. Okay.
Okay.
we
We
are
are
we
on.
are discussing we are discussing on this slides. So
Okay.
new source of genetic resource, because scientists have developed a new source of genetic resource and their conservation. So what type of the genetic resource? It means any types of the biological materials. Biological material, and then let us discuss on the recent in plant science, recent advance in plant science, because the recent advance in plant science science include the application of drone technology and GPS system for precision the agriculture, gene editing technique like CRISPR for develop the pest and disease resistance. So that's how we are discussing on the Pakistan. If we look into the Pakistan, because Pakistan is an agriculture country. Therefore, it is said that the agriculture is the backbone of Pakistan economy. About 80 to 85% population depend upon the agriculture directly and indirectly. If we look into the old of the agriculture, because old of the agriculture, our forefathers and our farming community driven by the pair of the blocks, and now the agriculture has replaced revolutionized the agriculture for the use of the application drone technology, GPS system, and global position system for use of the agriculture equipments in scientific methods, and the scientists have developed the pest and disease resistant and drought resistant, salt resistant, and improve the crops for the better understanding and managing the plant growth, pest and disease resistance, and improve the crop yield through the breeding and genetic modification to address the food security. Food security. Now, if we look into the global positioning system technology, because global positioning system technology used uh, either to track and manage the physical plant machinery, the how the primary tillage of the agriculture equipments and secondary tillage of agriculture equipments are used and guide the agriculture within an agriculture operations. GPS technology in agriculture enable the automatic machine and follow the precise the pathway ensure uniform application of resources, reduce the waste, and provide the real-time data in global or in real-time application resource waste and provide the real-time data for information graph management leading to improve the efficiency and higher. These are the big achievement of plant scientists, big achievement of agriculture science. They collectively work on this and revolutionize the in agriculture for increase the yield and for increase the yield of the different crops like sugar cane, like cotton, like wheat and other. So let us discuss on the global positioning. Next, please. Hello, hello. Next, please. That's how, these are the global positioning system. In the upper slide showed the global positioning system technology. These are the advanced agriculture the scientists have developed and you show look into the field. The field are the uniform in size and where is the next slides and showed that farming community driven by the pair of the blocks, and now the agriculture system here scientists have developed the various type of the agriculture equipment such as the combined harvest and others. So let us discuss on the biotechnology, because my first topic in the biotechnology. Bio means science and technology means because the technology used, the biotechnology is powerful and rapidly advancing field that use the biological system. Organisms are derived to develop the create, produce and technology that improve the human life and the health of the planet. Here's breakdown. It's impossible. So, and let us discuss on the molecular biology, the molecular technique in plant science, the molecular techniques use methods to study and manipulate the DNA, RNA, and protein to understand the plant genetic gene function and plant health and breeding. These are techniques include in DNA sequences, polymer chain reactions, PCR, identify gene as well as method for creating and usually the genetic modification in plants. In the molecular knowledge, that change the uh, we can add the foreign gene, add the foreign gene and remove the old gene from the crop and develop the uh, pest disease resistant, drought resistant, salt resistant crops, and these are all methods included in the genetic engineering. If we look into the genetic engineering, because genetic engineering is also called the modification of gene editing. Genetic means the study of transmission of character from parent to engineering. It means modification. Modification means engineering. Engineering it means the methods. How it modification? Because modification like through the CRISPR. CRISPR method is the process directly changing DNA of an organism to give it new traits or enable scientists to use it or add gene from one organism to another organism, putting bacterial gene into crops to make them resistant to pest, turn off edit gene to change how an organism grow and how the behave the create the genetic. Because genetic engineering is widely used in the medicine, agriculture, pharmaceuticals, and develop the insulin vaccine gene therapy, agriculture, genetically modified crops like golden rice and other very big innovations in the field of agriculture through the help of the genetic engineering. Through the help of the genetic engineering. Research studying disease testing. These pictures showed the genetic engineering. These are the DNA and deoxyribonucleic acid. It is present in the nucleus and cut the DNAs and foreign DNA, add the and remove the harmful DNA and others. And these are the methods. This is the methods to how to add the DNA, because DNA can be added through the process of creation of an insect resistant tomato plant. Insect resistant tomato plants. The how it what are the methods for develop of the creation of DNA. First of all, are the bacterium and the use of the bacterium DNA and insect resistant gene, the cut of the gene from the foreign gene cut through the molecular scissors, where the next step is the insert gene into the vector, because vector within the select vector is in a plasmid bacterium, an antibiotic resistant marker gene, and where is the antibiotic resistant gene. Number four, the copy vector in bacteria, and four copy target on gold are perfect within the DNA, and where the load vector coated particle tension and load the butter into the gene gun, and where is the shooting the gene gun referring to the particles at a high velocity of penetrating the plant cell. When the vector enter the cell, the gene are incorporated into the plant genome. Gene are the foreign gene are incorporated into the plant genome, and when the cell are plated on the selective antibiotic media, only cells that have incorporated the vector and will grow. And the next step, these cells are hello.
Yes, your voice is audible, dear Doctor
Uh,
Fahmi.
okay,
Can you continue?
okay, sir. Thank you very much. There is some problem of the net. Sorry, audience, and this is the uh, okay, and later we can discuss on the genetic engineering, because genetic engineering have revolutionized the various field and developed the various type and easy to the human life. The genetic engineering enables the development of gene therapies and precision the medicine tailored in the individual and the patients. And whereas the next stage, the vaccine development, the biotechnology was the key in rapidly develop the COVID-19 vaccine, mRNA vaccine, modern, and where and genetics engineering also play very important role in the pharmaceutical industry and disease diagnosis, production of biopharmaceutical development of insulin growth hormones, monoclonal antibodies, and other life-saving drugs are produced using through the genetically modified organism. Where the disease diagnosis and treatment, biotech advance improve the early diagnosis of the disease. If we look into the disease, the disease, and disease are there are the two types of the disease: biotic disease and abiotic disease. The biotic disease is incited by the different kinds of the microorganisms such as the bacteria, fungi, virus, nematodes, and other mycoplasma like organisms, where the abiotic disease is incited by different kinds of the environmental factors. So the genetic is a easy way to diagnosis of the disease. And genetically modified improve the nutrition of the crop. The genetically modified increase the yield, resistant pest disease. And if we look into our agriculture crops is facing different type of disease and pest, the genetical engineering has revolutionized it and developed the disease resistant genotype, disease and pest resistant genotypes, drought resistant genotypes, and salt resistant genotypes. These are the big issues in the Sindh particular in Pakistan. Where is the tolerance environment, drought resistant and pest, the improve the nutrition value, the biocrops like golden rice. These are the picture showed. The big issue of our biodiversity. If we look into the biodiversity, where the plants, animals are the living together. These are the in this planet. There are in the Pakistan. There are the two big issues. There. One is the climate change, and the other is the disease and the pest. Because climate change, big issues of our biodiversity. These are look into the issues of the biodiversity and climate change. If we look into the climate change, is one of the major challenges that agriculture will deal with in upcoming the years, both unpredictable weather events and occurrence of the biotic and abiotic stress of the disrupt the growing cycles of the grass, because climate change is one of the biggest issues in Pakistan. It puts negative impacts on our agriculture products on cotton, wheat, rice, as well as and fruit production, mango, date palm, pineapple, and so many. Where the different geographical regions threatening the for food productions, leading to the significant economic loss of major effectively use of plant genetic resources and innovative approach crop research are therefore the fundamental to ensure the global food security, because climate change and now the Pakistan and the Sindh particularly facing the issues of the climate change. The government and such organization give their priorities to give their priority to resolve this issue, where the biotic and abiotic states on the plants. Next, please, slides, please, sir. Biotic and abiotic issues on plants. These are the date palm. Date palm. Next slide. Big issues of our biodiversity, climate change, disease, and pest. Next, please. Next, please, sir. Biotic
Can we?
and about
Can you
next.
please number of
Yes,
slide?
next, please, sir. Yes, sir. Next, climate change. Uh, if we look into the climate change, because Pakistan is facing the big issues of the climate change, it puts a negative impact on the climate change on the our agriculture productions such as sugar cane, wheat crops, cotton crops, rice crops, as well as in the fruit production of Pakistan. The government and research organization give their priorities to burning this issue. The scientists have developed the different type of genotypes against the uh, uh, climate change. Back, yes, yes, yes. Yes, biotech. Yes, please. How climate change affect the biodiversity and agriculture? Agriculture. Because threats to the biodiversity in agriculture, because agriculture is the backbone of Pakistan economy. The climate change, biodiversity loss disrupt the essential services in the agro ecosystem due to the increase in temperature. For example, decline in pollinators. The pollinators play very important roles for the soil. The loss of the natural pest, because natural pest eating the in harmful insect, fungi, and others reduce the soil health. And third, two is the consequence of agriculture in Sindh. The vulnerable crops. Vulnerable crops, it means the attack of the crops, attack of the biotic and abiotic factors on the agriculture crops, such as the wheat, cotton, banana, intensely exposed to the climate-related biodiversity loss. The loss of resilience, decline in biodiversity make crops more vulnerable to pest disease and climate fluctuations. These are the laws of the climate change and environment. The picture showed the various type of the agriculture crops are infected by different kinds of disorder. These pictures are showed the negative impact of the climate change as well as disease. So, what plant scientists do for the food security crop improvement? So, it needs the I can clearly define the biotic, abiotic, negative impact, role of the plant biotechnology, role of the molecular biology, and genetic engineering, and old agriculture system, and global positioning agriculture system. Now, the what plant scientists do for the food security for our increasing the population. The crop improvement is the only method science practice of developing the new crop variety, enhancing the existing ones on to the make them more useful, productive, and resilient for combined traditional breeding methods within the modern molecular and biotechnological tools. These are the tools and the biodiversity conservation. Biodiversity conservation means the save of our animals, save of the plants, save of our planet, save of our earth. The plants, plant tissue culture, conservation, and food security, where is the crop improvement through the omics technology, genomes, omics, through the biotechnology, development, pest, pest disease and salt disease resistant crop through the molecular techniques. Where is the sustainable practice in agriculture? Because sustainable practice in agriculture are farming methods designed to the meet current food and fiber need without harming the environment, depleting natural resources. Dr. Saab, if we look into the Pakistan, because there are the various pesticide company are working and sell their pesticides because the sustainable agriculture practice use for the useful pollinators like trichogramma in sugarcane and biological control used in the cottons and agriculture integrate disease management practice used in the wheat crop and other crops. So these practices focus on the balancing economic viability, environmental health, and social well-being. Now the key sustainable soil health management. The crop rotation means the change of the crop, where the cover cropping, reduce or non-tillage farming, compost and organic manure use, the water conservation. How can we conserve the water through the drip irrigation and spring irrigation system, rainwater harvesting, where the scheduling of irrigation to reduce the waste, the integrated pest management, because integrated pest management used in the biological control beneficial insect microbes, where they increase the pest pressure in agriculture, rising the pest population, warming temperature accelerated the pest reproduction, allowing the more generation per season, expanded geographical range of warm climate pest invalid in new regions, where they increase the resilience, some pests become extreme weather and droughts. Need for the adaptive pest management, integrate pest management, where the combine biological, cultural, physical, and chemical controls, reduce the reliance on pesticide, promoting sustainable farming, flexible can be adjusted based on the specific pest climate change. So now these are the big issues in our agriculture crops and biotechnology and molecular and genetic engineering are used to easy then develop the different type of genotypes, and the lecture provides the various type of the how they conserve the genetic resource, how they collect the genetic resource, how they conserve it. The research and academic uh, research and academic guidance refer to the support, advice and direction provided to the student, scholar and researcher during the academic and research journey, help them in understanding the concept, improve the skills, and conduct an effective the research. Where the research guidance is assigning the identify the research problems, because we are the scientists, and we rely on the experimental research, not on the uh, uh, only the data collected. The two type of the research, scientific research, because scientific research conduct on the experimental research, going for the hypothesis, identify the problem, and then
Sorry
identify.
for inconvenience. Dear doctors,
Uh,
on which slide we are?
yes, sir. Research
Res-
guidance.
resistant guidance.
Research, research guidance. Next,
Okay.
next, next, next. And
Yes.
yes, yes. Research guidance. Research guidance is because we are the scientists, and we are the plant scientists. We rely only on the hypothesis and the experimental research. Experimental research identify the problems, and they conduct the experiment, and which type of the biotic, abiotic disorders in the plant, diagnosis process, through the conventional diagnosis process, through the lab diagnosis, and then going for the molecular test for identification of the problem. Advising on the research design, methodology, qualitative, quantitative, and experimental. Where the academic guidance, helping students choose the right course, subject, or specialization. Next, please, sir. Next, please. Hello. Advising on career paths related to the field of study. Next, please, sir. Yes. Uh, Education and capacity building, because global research institution and government working together to fund and share the innovative in plant science and biotech, knowledge exchange, training scientists, students and farmers worldwide throughout the joint the program and the seminar, the sustainability development goals, collaborative research projects aligned with security, climate action, and biodiversity, biodiversity, and finally the future collaborations. Fine, the future collaboration, the international seed banks, genomic database sharing, and genetic resources for global breeding efforts, where the public-private partnership join the venture between the university, biotech firms, and the NGOs to develop the scalable solution. Where the climate resilient innovation hub regional center focuses on the biotechnology solution for local climate challenges, and conserve the genetic resource and its conservation, use how it used in the local geographical regions than Sindh and Pakistan. So thank you very much. These are all kinds of the my lectures, and finally the acknowledgement. Acknowledgement. So first of all, we extend our heartfelt gratitude to the NOAA International Alliance for Science for their invaluable support and collaboration to organize the first international seminar on plant science. Their commitment to advancing scientific knowledge and focus on the international cooperation has played a role in making this event possible. Thank you very much, sir. I am highly thankful to NOAA International Foundation to give me a chance to share the scientific knowledge in the participant. Again, thank you, sir.
Thank you so much, dear Doctor Zulfikar Ali, uh, Wazir Ali Matla, and
Yes, sir.
we we honor to have you. Thank you so much for your precious time and
Thank you,
wonderful
sir.
presentation. It's
Thank you, sir.
if anyone have any question, you can write in inbox. We will give you an answer.
Yes, sir. I am available.
Uh,
Any questions? Hello.
write your question. If anyone
No.
have any question, you can write your question in chat box.
Okay, sir.
Can I ask a question?
Yes. Yes.
Okay. So, Sir Wazir, what do you think are the most common plant diseases that threaten crops in our region?
Madam, as far as my knowledge is concerned, the any abnormal occur in the normal health of the plants is called the disease, and there are the two types of the disease. One is the biotech disease, and the second is the abiotic disease. The biotech disease is incited by different kinds of microorganisms, fungi, bacteria, virus, nematodes, and abiotic disease is incited by different kinds of the environmental factor like environmental change in the climate change, moisture increase, and pH temperatures, pH temperature. Okay, madam.
Thank you
Hello.
so much, sir. Thank you so much,
Okay.
sir, for
Thank you. Thank you, madam. Okay. Anyone?
There is a question: is there any negative effects of GMO crops?
Is there a hello?
Any is there any negative effects of GMO crops?
GMO? Is there genetically modified organisms? So genetically modified organism graph increase the yield, resistance, pest, disease, and through the application of genetic engineering, use of the foreign gene to against the resistance, and there is the one example of the GMO crops, the golden rice. Golden rice and GMO crops is also used against the tolerant environment stage, drought resistant and pest resistant, disease resistant crops, and where the GMO genetically modified plants also play bifort crops like golden rice and coincide with the vitamin and minerals combined the malnutrition. Anyone, sir? Hello? Hello?
Here is a question: how to
Yes.
deal with biosafety of GMO crops?
Uh, Sir, first of all, the creation of an insect resistant plant, insect resistant in plants. There is the one of the example. So suppose one of the we can prepare the solution for the bacterium culture and the use of the plasmid, and the one is the insert the resistant gene. The resistant gene cut out the gene foreign gene from the through the molecular scissor, and where the insert genes into a vector within the selectable antibiotic and resistant marker genes. There is the they make their copy and coat within the uncertain gold particles within the DNA vectors, because vectors load the vector contain with the particle onto and the left end where the load filter into the gene gum and use of the shooting gene gum release the particles at a high velocity of penetrating the plant cells. Plant cell, the vector enter the cell, gene are incorporated into the plant genome. So plant genome where the cell are planted on the selective antibiotic and media. So media and yeah, we can use through the tissue culture and other MS mediums where they have incorporated in the vector and will grow. These cells are transport medium containing the plant growth factors and development of any plant who is the resist against the insect, pest, disease and other. These are the mechanism of GMO use and develop the new type of the resistance. Anyone, sir? Hello, hello.
Uh, Thank you so much
Uh,
for
and
your.
did you understand, sir?
Yes, yes.
Okay, thank you very much.
Okay.
Thank you.
Anyone have any question? You can write in chat box. So, if anyone have no question, then after the after the session, you need to submit your uh, insights on the session in comment box of the event. Okay, so it will be your final attendance. Yeah. So if anyone arise any question, uh, okay, after the session, you can share your question to me or any of our team member. We will provide. Yeah. The YouTube recording is available on YouTube complete, and on YouTube after the session you need to submit your feedback below the session of recording.
Okay, sir. Thank you very
So
much.
thank you so much, Dr. for your time and wonderful presentation. We honor
Thank
to
you.
have you, and hopefully we make international collaborations. Those are participants here from plant sciences field.
Okay, sir. Okay,
So
thank you. It's my honor.
soon we will work on it. So we want dedication and uh, passion from the researcher in plant sciences domain. I am repeating again. You need to submit your feedback end of the this presentation on YouTube. Recording is available. Okay, we will share the recording in group as well. Thank you so much, everyone. Wish you good luck and Allah Hafiz.